Hello there, this is Dr. Dan Guerra coming to you from Authentic Biochemistry Studios in the inland Pacific Northwest in an unusually warm afternoon. I'm not going to give you a weather report because we don't do that here at Authentic Biochemistry. Rather, we talk about biochemistry. Now, I am going to today, first of all, make this much smaller, talk to you about ceramide, one of my favorite sphingolipids, and its role in the largest sense, neuroendocrine aging. We're going to talk a lot of specifics here, so this is the broad brush topic that we're going to cover today. So let's just get started with this. Okay, so today is the 31st of January, and it is the year 2021. So what do I want to do in my first discussion here is go back to this paper published in Molecular Neurobiology. So let's go through some of this. I realize there's a lot of writing here. Don't worry, there are going to be a lot of really nice figures we're going to use today from some of these publications. So we know that ceramide, sphingosine 1-phosphate, are lipid mediators of the central nervous system for both development and dynamic retailoring of the connectome, including epigenetic phenomena. That's right, that's a lipid that's carrying out all these functions. So those two sphingolipids in particular, and intermediates between the two, are produced from neuronal and microglial membrane sphingomyelin. That's one biosynthetic route for ceramide and sphingosine. There are others, and I'll show you them, of course. And we've talked about them in the past. So you've got these sphingomyelins, and there's a constellation of cerebrosides as well in the central nervous system. And they're found non-randomly throughout the central nervous system and indeed the peripheral nervous system. So cell proliferation, differentiation, and cell fate are coordinated through chromatin remodeling, obviously, that's gene expression, and signaling, plus the post-translational alteration of membrane lipid raft domain, synthesis, movement within the cell, and in fact, turnover of those membrane rafts. Remember that ceramide and cholesterol and the right stoichiometric ratios are what make membrane rafts move. Right? Now, I want to remind you that neuronal apoptosis, autophagy, and various forms of death outright, like necrosis, and then the slow lack of division and ultimate death of the cell, senescence, also known as aging, are all within the panoply of potential cell fates. There are subdivisions of those too, which we'll talk a little bit about later on today. Eventually, they attain, over time, a dysfunctional transitional mechanism. So making those transitions in various cell fate signatures is something that each cell mass has to do throughout the lifetime of, say, a human being. But as people get older, like everything else that occurs in biochemical pathways and in physiological states, the regulation of those systems starts to break down. It does so because of the random introduction of mutations, but that's only one reason. Another is because of the actual senescent process, which basically hollows out the intense, complex regulatory systems that are overlain during the normal living process. And they drop out or they hollow out because the genes that are responsible for being expressed and the different isoforms of proteins and the different types of carbohydrates and lipids and nucleic acids and amino acid metabolic pathways that must occur concurrently in the cell for it to function correctly and deal with stress, all of that eventually starts to break down. And that leads to morbidity and ultimately to mortality of the system, obviously. Okay. So those transitional mechanisms break down during aging in a nonlinear chronological pattern. So, as I say, you have mutations or loss of sphingolipid metabolism enzymes, and that frequently leads to neuronal dysfunction and degeneration, or they are embryonically lethal. Okay, now that also can occur. Membrane rafts and exosomes plus free sphingolipid molecular species are secreted from CNS cell lineages and other cell lineages, of course, and they bind episodically to the extracellular matrix. Then they subsequently can be remobilized because of matrix metalloproteases, which are also secreted by neurons and microglia in the CNS, for example. And that allows for further sorting of these exosomes, binding to other locations. Cell surfaces is a common location. And of course, release into the circulatory system so they can move around and therefore encounter new cellular systems. Now, sphingolipids are secreted into the extracellular medium, and they also bind directly to cell surface receptors. They also interact with intracellular signaling pathways. They bind transmembrane domains of signaling proteins within the lipid bilayer, or they can produce transient membrane pores, even in various subcellular organelles like the endoplasmic reticulum, nuclear envelope, peroxisomes, the Golgi, the mitochondria, and all of that can impart a robust plenum of new functions which come and go during even a short period of time in the cell based on changes in metabolism, including bioenergetics and signaling, that can occur in very short time intervals. Okay, so that's kind of a background. That came from that paper. Now, continuing with this paper, ceramide serves several structural roles in organellar and in cellular membranes and in their microdomains, including multiple hydrophobic force-driven biophysical mechanisms of protein anchoring and assortment. Ceramide also directly mediates protein kinases as a lipid mediator, phosphatases, calcium binding proteins, ribosomal machinery, that means all the translation, secretory process that we just talked about with exosomes, and also porosomal dynamics, which we covered a couple of months ago, I think now, in how surfaces of cells allow for retailoring and restructuring of sections of the membrane to allow for movement and transport of very specific cargo into and out of the cell. Okay. It's a very important modification, porosomes, right? And again, we spent some time talking about those. You also have DNA repair associated with ceramide and heat shock protein movement, particularly in the endoplasmic reticulum and the Golgi. Ceramide-induced processes such as axonal degeneration, apoptosis, here we go back to cell fate, actually comprise both caspase mediated and caspase independent pathways. Those are the canonical and non-canonical apoptotic pathways. And they involve, of course, sometimes mitochondrial reactive oxygen that occurs because of electron transport chain and the lack of complete reduction of molecular oxygen to water. But it also involves things like P53, AKT, glycogen synthase 3 beta, which of course phosphorylates tau, which is involved in one of the phenotypes in Alzheimer's disease, or even the transcription factor activated protein 1, or AP1. Many of these we've talked about before in authentic biochemistry, now that I'm reciting these again from this lecture I just wrote. Now, adding to this florid involvement, the enzymes responsible for the synthesis of ceramide molecular species, of which there are several, like ceramide synthase isoforms, which I covered also in this lecture series, can alter the polarity and valency of ceramide agency. I know it's a mouthful, but I love that sentence because it covers most what you need to know in the broad stroke understanding of lipid-mediated biochemical alterations in the cell. Okay. Now, I promised you pathways. This is biochemistry, so here is a really good one. This is again from Molecular Neurobiology 2019. Okay. Remember that you have a de novo pathway for sphingolipids. Starts with palmitoyl-CoA, 16-carbon fatty acids, sterified thioesters, coenzyme A, plus the amino acid L-serine. Carry out this serine palmitoyl transferase reaction. Ultimately, two more reactions, one of which is 3-keto-sphingonine reductase, to make the next stable product, sphingonine. And then ceramide synthase, making dihydroceramide, then a desaturase, because you're putting in a double bond into that preformed palmitate. So you're basically making a trans palmitoleic acid directly in the sphingosine backbone of these lipids. Okay? And then you make ceramide, because that's a de novo pathway. Now, notice that pretty much goes from building blocks, palmitoyl-CoA and serine, to ceramide. However, the other ways of making ceramide can go this process. You can have a ceramide kinase reaction, which will phosphorylate ceramide, changing much of its valency, potency, and therefore agency in the cell. But you also have a phosphatase, which will strip off that phosphate and bring it back to ceramide. It's another way of reforming bioactive ceramide in certain subcellular locations in the cell, depending on what cell type. We haven't even started talking about receptors yet. And yes, ceramide has receptors. Ceramide can also be broken down can be used to make sphingomyelin, okay? So this is sphingomyelin synthesis, and we'll talk about how you make that, but basically it's adding a phosphorylcholine directly from glycerolipid metabolism, by the way. Trimethylated ethylamine, you know. And there's also sphingomyelinase, which we covered a great deal in the summer, in the fall, and early winter here at Authentic Biochemistry, both in the audio podcast and here on the video lectures. Now, sphingomyelinases can occur as a neutral or acidic sphingomyelinases, and that depends on where they're functioning in the cell. And they can generate directly from membrane sphingomyelin ceramide. Okay. Let's get it another way. 1, 2, 3. The last one is ceramide can be broken down via ceramidase, which means you're removing a fatty acid, one of the fatty acids that was added up here. Okay. And that's going to give you sphingosine. Okay. And there's also a ceramide synthase, which will build it back up. Sphingosine can be phosphorylated, just as ceramide can be phosphorylated. So there's the kinase and there's the phosphatase for that reaction. Now that's considered a salvage pathway. Now note that sphingosine 1-phosphate blocks ceramide synthase. So one of the branch point end products of this entire pathway can block the de novo synthesis of ceramide and therefore de novo synthesis of sphingosine base. Likewise, sphingosine 1-phosphate can inhibit ceramide synthase, okay, directly from sphingosine. Okay, so this product will inhibit the reutilization of sphingosine base to make ceramide. Likewise, once again, sphingosine 1-phosphate, not sphingosine-free base, will also inhibit the serine palmitoyl transferase reaction up here. So two different very important critical points of de novo synthesis are blocked by one of the branch point end products. Okay. Likewise, sphingomyelinase can also be inhibited both by the phosphorylated ceramide and the phosphorylated sphingosine. Multiple reactions are controlled at the level of end product inhibition. Okay, it's one way of looking at it. All right. Now, paper published in something called Biology Open, a journal that I've been getting more secure looking at, published in 2018, tells us the following. First of all, look at, you have a C2 ceramide, or this is a C6, there's a C2. That's how many carbons this fatty acid chain is Okay, this is coming from the sphingosine backbone. You're making ceramide, so you're adding a fatty acyl group here, right? So that's a C6. There's a C2 as an acetate. You can also have a dihydroceramide. I might as well put on these glasses, right? Where the double bond isn't there. See, that's a double bond. It's removed. So that's C2 dihydroceramide. Now, these three different ceramide isoforms, C6, C2, and C2 dihydroceramide, are used in the laboratory to cause inhibition of some of the reactions I just showed you. Okay, so they are very potent pharmaceuticals, basically. But then you have your normal substrate, when you find in the cell quite commonly, C16, where this is C16 fatty acid, palmitate again, C16 ceramide, very common isoform, molecular species of ceramide. Right? And of course, there's palmitic acid going through the biosynthetic pathway, and this is showing you that these reactions are occurring at the endoplasmic reticulum. It's really important to understand. These reactions are ER localized when you build sphingosine, sphingolipids in general. Okay. So, what is this long narrative going to say here? Since ceramide induces certain kinds of programmed cell death, such as apoptosis, it can also trigger macroautophagy. And that is basically just called autophagy in the literature. Its concentration in the cell, therefore, because it can tremendously alter cell fate, is very tightly controlled and regulated. So amino acid starvation, when that occurs, ceramide triggers autophagy by interfering with the mTOR signaling pathway and by dissociating a protein called Beclin-1, Beclin-2 complex in a C-Jun N-terminal kinase mediated Beclin-2 phosphorylation dependent manner. So you phosphorylate Beclin-2 and then you dissociate that complex of BCL1, BCL2. Okay, and the phosphorylation is conducted or carried out by the Jun N-terminal kinase 1. I want to show them there, right? So you get a subsequent stimulation of autophagy when that occurs. And it's been observed in various contexts in which the autophagy itself is increased because of long-chain ceramides. So we know that ceramide, particularly the C16 ceramide here, the common one found in the cell, will induce this system. Now, what is autophagy? Remember, the cell's under stress because it's basically under starvation or there's some other kind of stress that inhibits it from being able to take up nutrients from extracellular stores or utilize nutrients from intracellular stores. And that's because bioenergetics is being somehow reprogrammed in the cell. When this happens, the cell goes into a safety mode where it becomes autophagous, which means it starts digesting some of its large polymers, such as proteins, such as storage carbohydrate, if it happens to store glycogen, such as fatty acids, which it can store as triacylglycerol, for example. And it does this to reform new molecular species of pathway intermediates that are necessary for the new stress. So obviously the signaling has a tonicity to it and a telos to it. Right? Like everything that happens in biochemistry, it has a purpose once we've analyzed what's going on. It takes a long time sometimes to understand that purpose, and sometimes there's contrary and even looks like contradictory associated divergent pathways. But in the end, nothing is really contradictory because it simply works in a way that you get subcellular distribution of intermediates and you also get metabolic zonation, which allows for multiple things to be occurring almost simultaneously, but in different locations. Or not simultaneously, but differentiated hierarchically by time sequences and chronology. Okay. So it's notable that the conversion of short-chain ceramides like C2 and C6, shown up here, into the long-chain ceramides, which can occur just by swapping out this fatty acid, okay? When that happens, because of ceramide synthase, you don't get autophagy. So you need the long-chain ceramide molecular species for autophagy. So the dissociation of Beclin complex has also been observed in response to other pharmaceuticals like tamoxifen and a compound called PDMP, which is an inhibitor of the enzyme that converts ceramide to its glucosyl derivative, glucosylceramide. Okay? So drugs that increase the intracellular level of long-chain ceramides include those two pharmaceuticals. However, and in contrast to starvation, which is a common way to lead to autophagy, perhaps dysregulated autophagy, caused by the enforced dissociation from some other molecular mechanism of the Becklin complex, is related to the ability of ceramide to trigger not autophagy, but programmed cell death. Now, this was published way back in 2009. Okay, so this figure came from 2018, papers came from 2000. The reason I'm showing you both of these on the same slide is to give you an idea how science builds. That's how research works. Research is more important than calling it science, because research means as new techniques come about, and more importantly, as new ideas come about in cellular metabolism, in biochemical pathways, in pathophysiology, different disease states, new ideas come forth, new hypothetical deductions occur, and then new experiments are created to test yet those new hypotheses. Data is generated, evidence is finally derived from the data, and then a new induction occurs where we have, perhaps, if we're lucky, a sharper understanding of what's going on in nature, the nature of whatever system we're studying. So research is never a stay-in response. Research is never something that is a constant. It's always changing, and sometimes very rapidly, depending on the publication of a new seminal paper that comes out and blasts away all the previous work. And then, of course, that has to be reproduced sufficiently to generate enough trust that that new concept that is replacing the old concept is indeed one that we can rely on, at least for yet new research. These are really important concepts. I bring them up all the time, but they're not necessarily emphasized enough, particularly to physicians who are not scientists, right? They practice medicine. Some of them may be scientists in the laboratory, but most of them are physicians who do not practice science. And so what they read in the literature, if they do indeed read the scientific literature, may not be something that they understand as constantly changing over time. Okay? Because things can become fashionable in scientific literature, and then particularly people that use the scientific literature in biomedicine, they don't realize that fashionable reason for, say, thinking that cholesterol is some kind of toxin in the cell, which of course it isn't, that gets reified into the literature because the contributions to the literature try to presuppose that as a premise to their research. And then they try to keep on proving that same premise when indeed often it's not the case. And that's because the whole largesse of the literature would be the compendium necessary to really interpret the data from even one plenum of research, right, in one given area, let's say tumor biology. All right, so let's take a look at this slide here. You have a modulation of this pathway that you can see here is going to involve many intermediates we've talked about before. And notice that we start off with aging and neurodegeneration. So both of these pathophysiological states, aging is a pathophysiological state, and neurodegeneration, which can co-occur because of aging, such as Alzheimer's disease, will generate reactive oxygen. Now, reactive oxygen is going to have a modulatory effect on these kinases. This is sphingosine kinase, right? That's what that is there. So that's what that particular signature there means in this diagram that I got from molecular neurobiology. Okay. So, but notice that you can actually this same enzyme can actually function because of this synthesis of sphingosine 1-phosphate to decrease the amount of reactive oxygen. That's what this is showing here. Anyway, sphingosine 1-phosphate also can modulate phosphoinositol 1,3 kinase, which is an important signaling kinase cascade. But look what else happens here. Ceramide, on the other hand, which of course can generate free sphingosine via the ceraminidase, or can be formed from sphingosine via ceramide synthase, such as making that long-chain C16 ceramide. It can also be phosphorylated, but even before it gets phosphorylated, look at all the things ceramide does, and we've covered these already. But right now, we're introducing the fact that ceramide modulates the insulin growth factor receptor, okay? This is really important. This is a very significant hormone in the central nervous system, okay? IGF and binding to its receptor, okay? That's really important. Look how ceramide's modulating this insulin growth factor system in the brain. I know this is important because insulin plays a very small role in the brain, but insulin growth factor, which is a different protein altogether than insulin, has a very significant effect on signal transduction in the central nervous system. Okay, so along with that, ceramide will block, notice here, the phosphatidylcholine 3 kinase. Okay, so ceramide directly will block it, but ceramide 1-phosphate will activate this pathway. All right. Likewise, ceramide will block the AKT, so it's shutting off this pathway. Okay. So we have a modulation of phosphatidylcholine 3 kinase, AKT-mTOR signaling, which I mentioned just the last slide. And that occurs by many of these bioactive sphingolipids, which I just went through a little bit. Okay. And again, I'm telling you, this symbol here, when you see that, that means you have varied or ambiguous influence. Depending on the cell model used, the presence of reactive nitrogen species has also been found to inhibit, for example, downstream of this IKK beta. Okay, so there are multiple levels of regulation that this simple diagram is not showing you. Right? That's what we're saying here. So we have selected enzymes that you can see here. So in the sphingolipid metabolic pathway, we're not going to be naming all of them here, right? But keep a couple of things in mind. The scheme that's shown here is going to skip the inhibitory phosphorylation of this TSC, okay? That's the tuberosis sclerosis protein, right? So we're not going to show you any of that, at least not right now. And that's carried out by this IKK beta. Or this scheme does not also show influences of mTOR on the P13 kinase AKT itself. And the sphingolipids can modulate mTOR in many other ways, potentially allowing for cell types of cellular compartmental specific functioning of the sphingosine 1-phosphate mTOR branch, right, which we just were looking at here. Okay, we're going to get to that in a moment. All right. So, for example, mTOR1 down here, this is the complex with mTOR as its central kinase moiety. This is anabolic system, remember? mTORC1 activation occurs on the surface of various organellar membranes. So here's your subcellular zonation playing a role. The details of that pathway can be completely different than what's depending on different regions in the cell. Okay? You have to keep that in mind. All right. So when ceramide isn't blocking this pathway, but aging and neurodegeneration is carrying out this kinase reaction, and then processing this pathway in multiple ways. So sphingosine 1-phosphate working through receptor 3, isoform 3, is a positive effect on AKT. AKT will block TSC. Blocking TSC will prevent the blocking of REB, so REB can go on and activate mTOR. AKT directly from this sphingosine 1-phosphate cascade through that receptor lineage will also directly turn on mTORC1. mTORC1 will block the stress response, and in so blocking that will disinhibit, because of retrograde autophagy and mitophagy and also programmed cell death, will disinhibit what's going on downstream from this induction, or if you want to look at it, a turnover system for more neurodegeneration from the aging process. Okay? Because mTOR will de-suppress this pathophysiology. That's what this is saying. All right. Now, AKT will also activate IKK alpha. IKK alpha will turn on NF-kappa B. NF-kappa B will turn on mTORC1, and the same thing will happen. Further neurodegeneration, further aging. NF-kappa B itself will cause the production of more ROS and more reactive nitrogen, inflammation, and that's how you get this retrograde motion through this pathway. So it's a self-fulfilling prophecy. You have an aging system. There's some neurodegeneration which can be co-regulated, as shown down here, because of the senescence process, such as the senescence-associated secretory pathway or the Midas pathway we talked about. And then that will ultimately lead then to this entire carriage of a misjustice or an injustice to the central nervous system, right? Moving through this pathway, where ceramide and sphingolipids play a significant role, right? Differential role, and depending on whether or not they're phosphorylated. So, go on. Now I'm going to give you some more details about autophagy. There are a couple of different types of autophagy that can actually run into apoptosis. There is the stalled autophagy, which we'll cover first, where you start off with an autophagolysosome, which is induced, okay? And if there's further stress, you go from autophagosome, you pick up the lysosomal contents, you're going to start breaking down intermediate. Now, this is autophagy. You break down macromolecular structures, including organelles, right? That's mitophagy or peroxisomophagy, right? However, there is a possibility of this getting blocked. And once autolysosomal formation, this structure is degraded, you immediately go to default and you go to either apoptosis, which is a nice silent way of killing the cell, or necroptosis, which will, of course, spill out the contents of the dying cell. And when that happens, you get more inflammation. So this form of cell death can be mitigated by inhibition or genetic manipulation of cell fate, as we're showing you here. Although in these cases, cell death is autophagy dependent, it doesn't fit the definition of autophagy dependent cell death, which is different. For example, autophagy can be rerouted toward apoptosis or necrotosis, see? When it is activated at high levels, but it's defective in its last stages right there. And that's when it's fusing with the lysosome, or even during a process which involves autolysosomal self-degradation. Second, you can have a selective autophagic degradation. You start out with the autophagosome again. You induce stress, just like here. And then something is going on in this autolysosomal degradation pathway, right? You're starting to get cargo-specific receptors altering what's going on within this autolysosomal phagosome structure, right? When that happens, you differentiate into a different type of subcellular domain structure that will then degenerate to ferroptosis, frank apoptosis. This is one that involves iron. It's very toxic and inflammatory. Necrosis, direct necrosis, not involving a slow processing of cell death, right, which would be necrotosis, but necrosis, which is just immediate death. All of these can cause inflammation. So, example of such degradation includes targets including the KRAS protein, which sensitizes cancer cells to drug like tamoxifen, and that's owed to the reduction of MAP kinase signaling survival. There, the ROS, reactive oxygen scavenger catalase, which leads to ROS accumulation and necrosis, is what's being knocked down. The intercellular iron sequestering, there you go, protein ferritin triggering ferroptosis, and conversely, the transferrin receptor, which leads to reduced iron uptake, and then frank apoptosis. That's how these two are collinear, right, depending on the availability of sequestered iron. Okay. Autophagy-mediated degradation of inhibitors of death pathways has also been shown to activate directly apoptosis and various kinds of necrosis or necrotosis. Autophagy finally can have a misfunction in scaffolding. So here you have autophagosome induction, you have stress, but because of an alteration of the phagophore structure within this autophagolysosome, and because you have necrosomal structures forming associated with the RIP protein, the RIP kinase, or RIP, which means the death signaling, Or here's traditional caspase functioning leading to apoptosis. You get necroptosis, right? Necrotosis. Sorry, you don't mention that P anymore. So autophagy here can lead to cell death independently of its normal catabolic role. And here is shown the activity of the full pathway down to degradative steps isn't even necessary because the scaffolding function that's occurring here is happening within that phagolysosome, right? It's retailoring the way the proteins and the subcellular organelles are stacked together in a specific sort of structure. So alteration of scaffolding can automatically lead to caspase-mediated or RIP kinase-mediated cell death. That's what this is telling you here. These are structural modifications. All right. So there are three types of autophagy directed cell death. Remember, that's what that ADCD is. Excessive autophagy. This is death due to extensive self-consumption of organelles. Okay, so an over mitophagy and over peroxisomal phagy, releasing way too many hydrolytic enzymes and intermediates and pathways, right? And that's going to be a death-depending autophagy system, which will turn off the autophagy genes and turn on apoptosis. So autophagy, though, is required here. You have to start with autophagy in order to get to autolysosomal degradation. Okay? So excessive both autophagy resulting from massive induction of autophagolysosomes. Although membrane integrity is maintained, there's a ballooning of the perinuclear space. That's what occurs there. Excessive mitophagy alone, so only one of the organelles is being broken down. And this has to do with alterations in fusion phenomena of the mitochondria during the stress response. And so excessive mitophagy, autophagy-dependent selective elimination of mitochondria, that is going to also lead ultimately to death. And you're going to get a dramatic decrease in ATP. Why? Because your mitochondria are no longer functioning. So oxidative phosphorylation ceases, electron transport chain gets locked up, more reactive oxygen is made. Finally, you get autophagy. And autophagy involves changes in cell morphology. Okay, that's why we know it's distinct. And that includes, again, this PNS, this perinuclear space swelling now, and a focal rupture of the plasma membrane. So this is more like necrotosis or necroptosis. Right? If you get this swelling phenomena, it's because of water uptake. So death to the activation of sodium potassium ATPase, which is facilitating that water uptake. And you get a change, of course, in membrane osmolarity because of these changes in the ion channel passage, stoichiometry, as well as valency. And you get death-dependent autophagy, genes being generated, which turns into apoptosis. And here, autophagy flux actually isn't required because all you need is a swelling of the ER. So again, this is mitochondrial associated, ER associated. This is just the entire autophagolysosome up here. There's at least three different types, though, of autophagy-dependent types of cellular death. So in other words, leading down to the bottom of that hierarchy and killing the cell. All right. Now let's finish off a little bit more about this paper in Molecular Neurobiology. Mitochondria are produced by division, but irregular fission will corrupt their ontogeny. Okay? So non-symmetrical fission results in damaged, dysfunctional, or pathomitochondrial subcellular debris. That's what the result is. This imprecise fission product is then degraded via mitophagy. Okay? Mitochondrial fusion will allow for enhancing the size of naturally, physiologically potent mitochondria, and that will give you competent organelles. So fusion of mitochondria will give you still more competent organelles. Now, in a yeast model, where this was studied, sphingolipid intermediates increase with age, where they function to inhibit mitochondrial fusion. Now, that's not a good thing, right? That leads to fission and loss of bioenergetic competency, which can go down one of those pathways I just showed you to cell death. You see? So ceramide actually activates a dysfunctional fission of mitochondria, as an example, partially through the modulation of a protein called BUC, which is BCL2-related ovarian killer protein. It's part of the BCL2 complex we just talked about. Ceramide inhibits also AKT signaling. We showed you that in a slide or two ago. Synergistically with intracellular amyloid beta 42 or A beta 42, which is the famous protein, of course, for amyloid plaques or oligomers, more potently regulated towards Alzheimer's disease. So ceramide inhibits AKT signaling synergistically with intercellular amyloid beta 42, A beta 42, and that corrupts mitochondrial fusion-fission interaction relationships. And what that leads to is neuronal cell death, because it decreases the fusion-promoting proteins, mitofusin-1 and OPA1, which is optic atrophy 1 protein. Okay, so there, ceramide again is playing a very dangerous role. Ceramide also reduced the neuronal levels of the fission regulator dynamin-related protein 1, or DRP1, and that's a process reversed in muscle cells, which is interesting, presumably because of ceramide metabolism down that sphingosine pathway, for example. Maybe receptor expression, because you have multiple receptors for these ceramide molecular species, and even indeed sphingosine accumulation or sphingosine 1-phosphate accumulation. Okay, so here's another example of what's going on in the central nervous system in terms of regulation of ceramide causes one particular web of flow of events. It's the reverse in muscle cells, right? And even though it's the same compound being involved, the same molecular species even, because you have different receptors, or maybe you have different enzymatic pathways that are more robust in the muscle than they are in the central nervous system. Okay. Likewise, loss of ceramide transfer protein, known as CERT, which we talked a great deal about a couple months ago, results in disturbed transport of ceramides, of course, from the ER to the Golgi, where it likes to move towards to make those rafts. And then you get this hexacyl ceramide accumulation in the mitochondria, and a lower frequency because of that, because of this lipid there, of fusion and fission, which further then induces its mitophagy, which can result then in corrupted autophagy and then, of course, cell death. Lipid raft movement after ceramide biosynthesis also controls that tight mediation over mitochondrial and even frank cell fate. That's a process that deteriorates with aging and with any kind of stress like a cellular damage. All right. This basically shows you what I just mentioned here. You have this fission-fusion teeter-totter effect. Fusion generally healthy, inappropriate fission not. Ultimately, both of these processes involve some kind of new mitochondrial synthesis or mitochondrial biogenesis. Sphingosine bases generating ceramide. Ceramide then interacting with Bach to turn on fission. That's a bad fate for the cell. Ceramide can be converted to sphingosine 1-phosphate. That's going to lead to autophagy, which could include mitophagy, which is a good process. Ceramide can actually work through the LC3 protein directly for that. And ceramide through protein kinase A, the CREB, which is a transcription factor, PGC1 alpha transcription factor, and prohibidin 2, all then can lead to new mitochondrial biogenesis, either healthy mitochondria or fissionable material, means mitochondria are not really very functional. Likewise, sphingosine 1-phosphate turns on BCL2, which is, of course, triggering a potential for apoptosis, but it blocks BAX, BAD, and BIM. Okay? BIM actually is another chaperone protein for the mitochondria. And the production of BCL-XL, of course, blocks ceramide utilization. Here's where OPA and mitofusion is involved, because ceramide blocks that, you don't get healthy fusion. So this just reiterates what I talked about before, all from that same paper. Now, here's the payoff for all of this. Ceramide sphingosine 1-phosphate, could be interconverted. And neurodegeneration generates more ceramide. Ceramide can cause mitochondrial dysfunction, the processes I just explained to you, the events I just explained to you. The A beta protein metabolism, tau hyperphosphorylation can lead to tau fibrillary tangles. This is all Alzheimer's pathophysiology, Alzheimer's pathophenotype. So you get pro-apoptotic signaling from ceramide. So ceramide is a lipid which brings on a more potent and florid Alzheimer's disease. And that's why it's iterated here as neurodegeneration. Sphingosine 1-phosphate will generate an immune response and can actually cause survival signaling. This is the autophagy pathway. And that includes P13 kinase, AKT, and that BCL2 family, all of which is going to lead to autophagy rather than to programmed cell death. So the increase in ceramide at the expense of sphingosine 1-phosphate enhances degeneration. Okay, that's what's going to occur here, ultimately, because it inhibits the phosphatidylinositol 1,3 kinase AKT survival signaling via the modulation of the A-beta protein and the sphingosine 1-phosphate receptor dependent immune response, which is shown down here. The latter capable of exerting either beneficial, that is AB clearance, so it's a potential therapy, an intact internal cogent therapy to slow the process of Alzheimer's disease. Or you can have a detrimental outcome, which can cause a damage to neurons. That's also going to occur because this modulation of neurodegeneration has not been well understood. Probably has to do with receptor-mediated responses and interconversions, as we talked about. All right. So again, remember you've got stress. Stress can give you a cytoprotection. That's the primary response, giving you autophagy. Autophagy, if it doesn't go through one of those pathophysiological pathways because of ceramide, will block apoptosis and oncosis. However, prolonged stress will lead to a point of no return, and that'll lead to all those different autophagic cell deaths, which we just talked about. And that's the fail-safe response, because when you get to this place, in this stage of cell alteration, the cell has to kill itself. So I'm going to stop here because I think it's a good place to stop. Next time we get back together, I'm going to start talking about mTORC in more detail. Okay, but right now I want to end today's lecture and wish you a very happy what's left of the weekend. And tell you that this is Dr. Dan Guerra from Authentic Biochemistry Studios on the 31st of January, saying bye for now.